Antihelminthics or Anthelmintics are a group of antiparasitic drugs that expel parasitic worms, also known as helminths, and other internal parasites from the body by either stunning or killing them and without causing significant damage to the host. They may also be called vermifuges, those that stun the parasites, or vermicides, those that kill them. Helminths are classified into roundworms, also known as nematodes, and flatworms. As an example of nematodes, we have Ascaris lumbricoides. Parasitic nematodes cause substantial morbidity and mortality in animals and people globally. Flatworms are further classified into trematodes, also known as flukes, for example Schistosoma mansoni, and tapeworms, also known as cystodes. As an example of cystodes, we have Tinea solium. Trematodes are flattened, oval, or worm-like animals. Their most distinctive external feature is the presence of two suckers, one close to the mouth and the other on the underside of the animal. Cystodes have no gut or mouth and absorb nutrients from the host's alimentary tract through their tegument, through which gas exchange also takes place. Worm infection is also known as helminthiasis. Drugs used to treat helminthiasis, also known as antihelminthics, are classified according to the group of parasites that they affect. So we have antinematodal agents, antitrematodal agents, and lastly anticystodal agents. In this video we'll discuss the mechanism of action, indications, and the side effects of two very commonly used antihelminthic drugs, namely albendazole and mebendazole. Albendazole and mebendazole are primarily antinematodal drugs. Both of them are effective against Ascaris lumbricoides, which causes ascariasis, Enterobius vermicularis, also known as human pinworm, which causes enterobiasis, and Trichus trichiura, also known as whipworm, that causes trichuriasis. Albendazole is also effective against Ancylostoma duodenale, also known as Old World hookworm, and Necator americanus. Ancylostoma duodenale and Necator americanus are the two human hookworm species that are normally discussed together as the cause of hookworm infection. Albendazole and mebendazole also can be effective against some cystodes, for example Echinococcus granulosus, which causes hydatid disease, Tinea solium, which causes cysticercosis and neurocysticercosis. Both agents are effective against Tinea saginata, which causes tinnitus. In this picture you can see the cross-section of Ascaris lumbricoides. It consists of body cavity, intestine with epithelial cells that absorb glucose, amino acids, and other nutrients, muscle cells that contract and move parasites' body, oviduct, and uterus. All nematodes have a similar structure. Albendazole and mebendazole will disrupt cellular processes in two types of cells, epithelial cells in the intestine and muscle cells. In the image in the right corner, you can see the intestinal lumen, intestinal villi, crypts, and those tiny intestinal epithelial cells, also known as enterocytes. Let's zoom inside one nematode cell. Specifically, we care about intestinal and muscle cells of the worm. Because drugs will inhibit important functions in these cells, this will kill or stun the parasite. Here you can see the cell membrane. In the cell membrane are glucose transporters type 2, GLUT2, that enable protein-facilitated glucose movement across the cell membrane. A part of glucose is transformed into glycogen, process known as glycogenesis. Glycogen can be broken down into glucose, process known as glycogenolysis. Okay, now let's review a tiny bit of biochemistry. During glycolysis, glucose ultimately breaks down into pyruvate and energy. A total of 2 ATPs is derived in the process. Pyruvate is converted to acetyl-CoA. Then we have Krebs cycle. The reactions of the cycle are carried out by 8 enzymes that completely oxidize acetate in the form of acetyl-CoA into 2 molecules, each of carbon dioxide and water. For each glucose that enters glycolysis, products of the citric acid cycle are 2 ATPs, 6 NADHs, and 2 FADH2s. The largest quantity of ATP is formed through oxidative phosphorylation in mitochondria. All the NADHs and FADH2s constructed in previous steps are used to synthesize ATP. Okay, now let's review a tiny bit of molecular biology. Tubulins are globular proteins. There are two types, alpha and beta tubulin. Alpha and beta tubulins polymerize into microtubules, a major component of the eukaryotic cytoskeleton. Microtubules function in many essential cellular processes, including mitosis. On the beta subunit, there is a specific domain called the colchicine-sensitive site. As a side note, colchicine is a classical antimitotic drug that blocks mitotic cells in metaphase. It binds to soluble tubulin to form tubulin-colchicine complexes in a poorly reversible manner, which then binds to the ends of microtubules to prevent their elongation. If you stop the elongation of microtubules, you stop mitosis. It's simple. Now, why am I analyzing this in such crazy detail? You'll see in just a second when we discuss our antihelminthic agents. Alpha and beta tubulins polymerize into dimers, then dimers polymerize into microtubules. As vermicides, albendazole and mebendazole causes degenerative alterations in the intestinal cells of the worm by binding to the colchicine-sensitive site of beta tubulin, thereby blocking polymerization of tubulin dimers in intestinal cells of parasites, thus inhibiting its assembly into microtubules. Disruption of cytoplasmic microtubules blocks the uptake of glucose and other nutrients. Why? Well, it sounds a little strange maybe, but microtubules have a huge role in glucose uptake. When microtubules are damaged, glucose uptake is low or absent. We can say that albendazole and mebendazole inhibit glucose transporter type 2, GLUT2, indirectly. It's worth mentioning that albendazole binds much better to the beta-tubulin of parasites than that of mammals. That's why it's very toxic for the parasite cells and less toxic for human cells. Okay, so because glucose uptake is low, the level of glucose inside the cell is also low. Impaired uptake of glucose by the larval and adult stages of the susceptible parasites depletes their glycogen stores, because glycogenesis is nearly absent. If the level of glucose is low, the glycolysis decreases until eventually it stops. What about those two ATPs from glycolysis? Will they be synthesized? Of course not. Will the cell form new pyruvate molecules? No. What about Krebs cycle? It will be inhibited also. Will the cell synthesize 2 ATPs, 6 NADHs, and 2 FADH2s? Of course not. The parasite won't form new ATP via oxidative phosphorylation. The level of ATP inside the cell will drastically decrease, and there are bad consequences for the parasite cell. The sodium-potassium pump, for example, is a form of active transport that requires energy in form of ATP. It moves 2 potassium ions into the cell where potassium levels are high and pumps 3 sodium ions out of the cell and into the extracellular fluid. Without ATP, the pump would cease to function, which will lead to electrolyte imbalance. In the absence of ATP, all cellular processes, like protein synthesis, will stop. Parasites' muscle cells will not contract, which will lead to paralysis. The parasite will be immobilized. Cell motility will also decrease. Because microtubules are damaged, maintenance of cell shape and intracellular transport are also disrupted. All this will lead to the parasite cell's death and finally the parasite's death. Albendazole also prevents the formation of spindle fibers needed for cell division, which in turn blocks egg production and development. Existing eggs are prevented from hatching. Some parasites have evolved to have some resistance to albendazole and mebendazole by having a different set of amino acids that form the colchicine-sensitive site of beta-tubulin, decreasing the binding affinity of albendazole and mebendazole. In the picture, you can see 3 new amino acids that make up a slightly different colchicine-sensitive site. That's why the binding affinity of these two antihelminthic agents decreases in parasites that have mutated beta-tubulin. The liver, testicles, and gastrointestinal tract are the most affected organs after exposure to high albendazole and mebendazole doses. But why are these drugs toxic, you'll ask me? Because our cells also have beta-tubulin and microtubules. The structure of human beta-tubulin is slightly different. That's why we don't die too. But still, albendazole and mebendazole will bind to it and cause those toxic effects that we previously discussed, but in a decreased manner. Also, a big reason why people don't die together with the parasite is that they're much bigger. The most common side effects of albendazole and mebendazole are headache and abnormal liver function. The liver enzymes normally increase 2 to 4 times the normal levels. An estimated 1 to 10% of people experience abdominal pain, nausea or vomiting, dizziness or vertigo, increased intracranial pressure, temporary hair loss, and fever. In rare cases, it has been associated with a dangerously low white blood cell count, low platelet count, and a risk of agranulocytosis. This concludes our lecture. Thank you for watching.